In the thermal power plants, steam created by burning coal spins the turbines of the generators that create electric current. While electric energy is being created, oxygen is consumed and carbon dioxide is created. In a similar way, CO2 is created by burning carbon-based organic molecules in the human organism, while oxygen is consumed in oxidative phosphorylation and billions of mitochondria nanogenerators called ATP. That is why the regulatory mechanisms of the organism have to arrange a constant supply of the required amount of oxygen from air to the cell mitochondria and the drainage of the carbon dioxide from the organism to the atmosphere. We give out energy continuously, but we accept it from our surrounding irregularly while eating in the form of food. The whole complexity of the regulation of our metabolism is caused by these facts. Among other things, it is necessary to regulate the generation and breakdown of the energetic supplies, such as the fast accessible glycogen in the liver and the supplies of energy in the form of fat in fat cells. Let us detach ourselves from the complexity of the regulation of metabolism and focus on the basic principles through which the cells gradually gain energy from ingested food, use oxygen, create carbon dioxide, and in the end give rise to the universal energy carrier in cells, the ATP. A vast majority of our food contains proteins, polysaccharides, and lipids. Before the cells can use them as a source of energy, they have to first be broken down to simple subunits. Proteins to amino acids, polysaccharides to simple sugars, and fats to glycerol and fatty acids. Therefore, the first part of the energetic transformation of food is digestion. Connected with this depolymerization, it takes place outside the cells of the digestive tract or inside the cells in specialized organelles, lysosomes. After the macromolecules have been pre-digested, little organic molecules derived from food enter the cytosol of the cell, where their gradual oxidation starts. The second phase of biological oxidation starts in the cell cytosol and finishes in mitochondria, which could be called energetic centers of the cell. The glucose molecule gradually transforms in a series of chemical reactions called glycolysis, and finally is split into two molecules of pyruvate. In a similar way, other sugars are transformed into one of the sugar intermediaries of the glycolytic metabolic pathway and are also finally made into pyruvate. During the creation of pyruvate, two types of activated carrier molecules arise, ATP and NADH. ATP, as a universal carrier of energy in the cells, arises here without the need to consume oxygen. However, carbon dioxide is also created during the glycolysis, and it is necessary to expel it from the organism. NADH goes to mitochondria where it is needed for oxidative phosphorylation, during which ATP is created from ADP while consuming oxygen and creating water. Pyruvate can also be created by transformation of some amino acids. Pyruvate goes into mitochondria where it yields carbon dioxide while it is being transformed to an acetyl group connected to coenzyme A. Thus, acetyl coenzyme A is created. In addition to this, a large amount of acetyl coenzyme A is created during the gradual oxidation of fatty acids that were originally a part of fats and is also created by transformation of some amino acids. In the third phase, which takes place exclusively in mitochondria, all the acetyl coenzyme A is gradually oxidized. The acetyl group from coenzyme A enters a chain of reactions called the citric acid cycle. Some amino acids can also enter the cycle. Ammonium groups are dissolved from the amino acids as ammonium ions. Ammonium ions are generally transformed into urea in the liver and then eliminated via urine. Alternatively, they can be transformed to the molecules of glutamine, which are metabolized in the kidneys, while ammonium ions are recreated and eliminated into urine. The result of this cycle chain of reactions of the citric acid cycle is the production of CO2. At the same time, the cycle is a source of a large amount of energy-rich carriers of electrons called NADH, and on a smaller scale also of the similar FADH2. The NADH and FADH2 molecules are a fuel for the mitochondria energetic nanomachines, which in mitochondria nanogenerators produce ATP, while at the same time consuming oxygen and creating metabolic water. Since the energy for fueling the ATP synthesis generally comes from oxidative breakdown of food molecules, the phosphorylation of ADP to ATP is called oxidative phosphorylation. All the energy from food is in the end transformed to universal energy packages of ATP that contain chemical energy in the form as phosphor anhydride bond. Whenever energy is needed in the cell, it can be yielded by cleaving this high energy phosphate bond of ATP. During the process, ATP is split into ADP and phosphate. The overall efficiency of the ATP production process is great. Almost half of the energy which can be theoretically obtained from the oxidation of glucose or fatty acids to water and CO2 is harvested and used for the energetically unfavorable binding of the phosphate to the ADP molecules. The rest of the energy is released in the form of heat. The involved nanomachines are much more efficient than, for example, a car engine, which is only capable of transforming a bit over 20% of the energy available from the fuel into useful energy. So, to summarize the whole process in a simple way, we could say that the food molecules are broken down in 3 phases, and in the end they give rise to the universal carrier of energy in the cells, ATP. Production of the ATP itself takes place in mitochondria, the energetic center of the cell. Mitochondrion is separated from the surrounding by 2 membranes, inner and outer. These membranes divide the mitochondrion into 2 compartments, a large inner space called the mitochondrial matrix and a narrow intermembrane space. The outer membrane is relatively permeable for dissolved small molecules because it contains many water channels going from one side to another. These channels are formed by specialized proteins, porins. On the contrary, the inner membrane of the mitochondrion, analogously to the cytoplasmic membrane of the cell, is impermeable for ions and most small molecules, which can only be taken across by membrane transport proteins. Also, its inner side has a negative electrical potential. The inner mitochondrial membrane is arranged into many folds. The arrangement significantly enlarges its surface area. It is important since this is where the nanogenerators that create ATP are placed, on this inner membrane. The principal fuel for the production of ATP is the energy accumulated in the chemical bonds of NADH and FADH2 molecules. As explained previously, this energy comes mostly from the citric acid cycle, through which all the food molecules were broken down. The molecule of the reduced nicotinamide adenine dinucleotide, often abbreviated as NADH, sheds away a hydrogen ion and passes the accumulated energy on for further usage. The energy is given away in the form of 2 high-energy electrons. Nicotinamide adenine dinucleotide, abbreviated as NAD+, then leaves for another load of energy that it obtains from the chemical metabolic reactions coupled with the reduction of NAD+ to NADH. In a similar way, the molecule of the reduced flavine adenine dinucleotide, often abbreviated as FADH2, also passes energy on through 2 electrons with high energy while shedding 2 hydrogen ions. The molecule of FAD then receives another package of energy in one of the reactions of the citric acid cycle and is reduced back to the molecule of FADH2. The energy contained in the electrons passed to the mitochondria energetic complexes by the molecules of NADH and FADH2 is used for pumping hydrogen ions from the mitochondrial matrix into the intermembrane space of the mitochondrion. Positively charged hydrogen ions, protons, are, because of this energy, actively pumped against the direction of the electric potential. The inner side of the membrane has negative potential, and the active pumping further heightens this potential. After passing on the energy, the electrons are at the end utilized in the binding of the oxygen and hydrogen ion, which together form a molecule of metabolically produced water. Contrary to the burning of the hydrogen, where all the energy is released explosively in the form of heat, the energy in biological oxidations is transformed gradually in a step-by-step fashion. The passed-on energy of the electrons accumulates in the electrochemical gradient of the hydrogen ions built up across the inner mitochondrial membrane. The concentration of the hydrogen ions in the intermembrane space of the mitochondria is about 10 times higher than in the mitochondrial matrix. Since the ions pass through the outer mitochondria membrane easily, thanks to the water channels, their concentration in the intermembrane space is almost the same as in the cytosol. The combined power of the concentration and the electric gradient pushes the hydrogen ions across the inner membrane of the mitochondrion back to the matrix, whilst spinning the rotors of the nanogenerators of the ATP synthase. As unbelievable as it seems, these are true nanogenerators that are propelled by the current of the returning hydrogen ions. As the nanogenerators spin, the cam attached to their shaft mechanically changes the spatial conformation of the immobile stator. This change in conformation helps to create the chemical bond between the molecules of ADP and phosphate, that is, the ATP synthesis. Let us look at the operation of this very efficient molecular biological power plant in more detail. A chain of 3 protein complexes built into the inner mitochondrial membrane passes over the electrons from the molecules of NADH and FADH2 gradually to one another, uses their energy for pumping hydrogen ions, protons, and finally builds these electrons into the produced metabolic water. The names of these 3 protein complexes are NADH dehydrogenases, cytochrome BC1 and cytochrome oxidase, respectively. There are 2 other mobile carriers, ubiquitin and cytochrome C, that also constitute the electron transport chain. NADH passes its 2 electrons first to the complex of NADH dehydrogenases. This complicated protein enzymatic complex is enabled to utilize part of the energy from the passed over electrons for pumping hydrogen ions. The energy of 1 electron is used for pumping 2 hydrogen ions out. Each NADH molecule therefore contributes to pumping 4 protons through this complex. 2 electrons are then passed over to the mobile molecule of ubiquitin. This molecule passes both the electrons to the second complex, which is called cytochrome BC1. Cytochrome BC1 uses the next part of the energy of the accepted electrons, again for the transport of hydrogen ions. Energy from 1 electron is sufficient for pumping 2 hydrogen ions out. 2 electrons from the NADH molecule therefore ensure pumping of 4 other hydrogen ions. Each electron for the cytochrome BC1 is passed to the mobile carrier cytochrome C. Cytochrome C carries only 1 electron at once. Cytochrome C passes the 2 electrons one by one to the last complex in the chain, the cytochrome oxidase complex. Gradually, 4 electrons get together here. These electrons interact with one molecule of oxygen and with 8 hydrogen ions. 4 electrons, 4 hydrogen ions, and a molecule of oxygen form 2 molecules of water. The remaining 4 hydrogen ions are pumped over the inner mitochondrial membrane. All 3 complexes operate simultaneously, as long as they have a sufficient supply of electrons. They pump hydrogen ions while they gradually pass the electrons from one to another, using ubiquinone and cytochrome C as carriers. The source of electrons from the electron transport chain is not only NADH, the molecule of reduced flavine adenine dinucleotide, FADH2, can also provide them. However, this molecule skips the complex of NADH dehydrogenases and it passes the electrons directly to ubiquinone. ATP synthase is a miniature biological engine propelled by the hydrogen ions that are returning to the mitochondrial matrix, driven by their electrochemical gradient. The nano engine has a large head that functions as an immobile stator and is linked with the membranous part of the assembly by a stalk. On the outer side of the stator, there are binding places for the ADP and phosphate, alpha and beta subunits. The inner spinning part, the rotor, consists of a ring of protein subunits buried inside the membranous part of ATP synthase. This ring forms an inner wall of narrow channels located on the interface between the stator and the rotor. As the electrons squeeze their way down the electrochemical gradient to the mitochondria matrix, they spin the rotor. Part of the rotor is a shaft protruding into the stator part. The shaft ends with a cam, whose spinning periodically changes the spectral conformation of the stator subunits in such a way as to help forming of the covalent high-energy bonds between the phosphate and the ADP molecule. It is in this way that the energy accumulated in the electrochemical gradient of hydrogen ions is transformed to the mechanical energy of the spinning rotor with the cam. This mechanical energy is then transformed into chemical energy of the covalent phosphate bond and the resulting ATP molecule. 3 molecules of ATP are produced in one spin of the rotor. The ATP synthase is able to produce more than 100 molecules of ATP per single second, or 33 revolutions per second. However, the spinning speed depends on the magnitude of the hydrogen ion gradient. The number of hydrogen ions needed for spinning the rotor for one complete revolution is about 12. Yet, it still depends on the type of the ATP synthase. It varies between 10 and 14. The energy of 2 electrons from one molecule of NADH is sufficient for pumping 4 hydrogen ions across by NADH dehydrogenases, 4 other ions by cytochrome Bc1, and 2 more hydrogen ions by cytochrome oxidase. Therefore, 1 molecule of NADH is sufficient for pumping 10 hydrogen ions across the inner mitochondrial membrane in total. In one revolution, the ATP synthase produces 3 molecules of ATP. If 12 hydrogen ions are needed for one revolution, then 10 hydrogen ions that have been pumped using the energy of 1 molecule of NADH produce on average 2.5 molecules of ATP. The energy of 2 electrons from 1 molecule of FADH2 is sufficient for pumping only 6 hydrogen ions across the membrane, 4 by cytochrome Bc1 and 2 by cytochrome oxidase. The energy released from 1 molecule of FADH2 is therefore only sufficient for producing 1.5 molecules of ATP. The ATP synthase is not the sole process which benefits from the electrochemical potential of the hydrogen ions. The influx of the hydrogen ions to the mitochondrial matrix is also coupled with the transport of various substances across the inner mitochondrial membrane. For instance, pyruvate is carried to the mitochondrial matrix together with the hydrogen ions. Pyruvate, after its transformation to acetyl coenzyme A, functions as fuel for the citric acid cycle. In a similar way, the electrochemical gradient of the hydrogen ions drives the influx of inorganic phosphate that is necessary for the ATP synthase. The electrical potential of the inner mitochondrial membrane also drives the coupled transport of ATP and ADP. The ATP molecule has 1 more negative charge than the ADP molecule. Therefore, the gradient of the electric potential pushes it out of the matrix in the process of its exchange for the ADP molecule. The transport of blood gases to the cell is driven by concentration gradients. The consumption of oxygen creates a concentration gradient between the mitochondrial matrix, the cytosol of the cell, the interstitial fluid surrounding the cell, and the oxygen-bringing capillary. In a similar way, carbon dioxide produced mainly in the citric acid cycle is carried along its concentration gradient from the mitochondrial matrix to the cell cytosol, and from the cell cytosol through the interstitial liquid to blood. If the consumption of oxygen increases, the concentration gradient between the mitochondrial matrix, cell cytosol, interstitial liquid, and the capillary also increases, which results in higher oxygen inflow to the cell. However, the situation of higher oxygen consumption also decreases the levels of oxygen in the capillary. This is the reason why the physiological regulatory mechanisms also provide higher oxygen supply through the capillary. The consumption of oxygen depends on the rate of ATP production. An increased rate of ATP synthase raises the consumption of oxygen in the mitochondria. Yet, the consumption of oxygen is not governed directly by the concentration of oxygen in the mitochondrial matrix. It is governed by other factors, for instance, the ratio between the ATP and ADP concentrations. If the concentration of the oxygen dissolved in the mitochondrial matrix drops below a certain critical level, for instance, due to inadequate blood perfusion, the supply of oxygen to the cytochrome oxidase does not suffice the demand of the final water-producing reaction between the electrons, hydrogen ions, and the now scarce oxygen. The production of metabolic water and the consumption of oxygen decreases. The passage of electrons through the electron transport chain also slows down, resulting in a gradual lowering of the hydrogen ion gradient across the inner mitochondrial membrane. The driving force of the ATP synthase turbine diminishes and the ATP production decreases until it ceases completely. A shutdown of ATP production can also occur in cyanide poisoning because cyanides block the activity of the cytochrome oxidase. In this circumstance, mitochondria can be full of oxygen, but they are not able to utilize it. Oxygen is brought to the tissue and the cells by circulating blood. Blood also carries away the carbon dioxide produced during the metabolism. In blood, oxygen is transported bound to hemoglobin inside red blood cells. Oxygen enters blood and carbon dioxide leaves it in lungs. The transport of oxygen and carbon dioxide inside the respiratory system is governed by partial pressures of oxygen and carbon dioxide in the inhaled air. The mutual dynamic relationship between the composition of the inhaled air, barometric pressure, the activity of the respiratory system, the concentration of hemoglobin, the function of the circulatory system and the oxygen consumption, and carbon dioxide production in the metabolism can be studied with the aid of our mathematical model.